Hi, my name is Nur Safia binti Lalim with metric number 145873. In this video, I will present about the aseptic technique in terms of microbiology and most importantly, to explain about how to use the proper aseptic technique to transfer the bacteria from one agar plate to another agar plate. There are 5 topics that I will discuss and explain about, which is the first one is introduction, where I will talk about the principle of the aseptic technique. Second is the objectives. Third is the procedures, where I will talk about the steps involved in this aseptic technique experiment. Fourth is the precaution to make sure that we can minimize the possible error that can affect the results. And lastly, the conclusion, where I will discuss about the overall overview of these procedures. So now I will start with the introduction. Aseptic technique is actually a technique used to prevent the contamination of cultures from the foreign bacteria contained in the environment. It is very important to prevent the culture from being contaminated because it may later affect the result. This aseptic technique is a technique that is very important for the microbiologists because it is widely used for variety procedures such as transferring cultures, inoculating media, isolation of pure cultures, and also when performing microbiological tests. Besides that, the importance of the aseptic techniques is also to prevent the microbes used in the laboratory from accidentally being released to the environment or even infecting people who is working in the laboratory. Other than that, aseptic technique is essential for the isolation of a single species of microorganism from the mixed culture to obtain the pure culture. Furthermore, it is also important to maintain the pure stock cultures while transferring the culture to the new media. This is the objectives of the aseptic technique, which is first is to acquire the skill of aseptic technique in the field of microbiology. Second is to prevent contamination of cultures and media from microbes in the environment. Third is to transfer cultures from one medium by inoculating another medium. Fourth is to isolate a microorganism from a mixed culture to obtain a pure culture. Lastly is to prevent lab microorganisms from being spread in the environment and infecting the investigator. Next, I will talk about the procedures of how to transfer the bacteria from one agar plate to another agar plate. Before that, let me introduce the materials needed in this experiment, which is the mixed culture of bacteria on an agar plate labeled with A, another agar plate labeled with B, inoculating loop, agar gel, Bunsen burner, and table disinfectant. So the first step is always the preparation of the work area. We have to make sure that the workspace was cleared from all non-essential items, and the table must be cleaned using the disinfectant. This step is important to kill all unwanted bacteria and so decrease the chance of the agar plates becoming contaminated. Next, the agar plate B needs to be outlined with 4 sections from A to D on the bottom of the agar plate as shown in this figure. This will make the quadrant strip method to be easier to be done later. Then flame the loop until it is red hot and let it cool down. This step is important because it will sterilize the loop and make sure to kill any unwanted bacteria that are present on the loop. After that, obtain a loop full of the mixed culture from an agar plate A and put that loop full of bacteria into the section A of an agar plate B to make a successive streak on the agar surface. This allows the bacteria to spread out and to grow in individual colonies on the agar plate. Then flame the loop again and let it cool for a while. Again, this step is important to make sure that the loop is sterilized and kills any unwanted bacteria that are on the loop. Next, by using the sterilized loop, streak on the agar surface in the section B. Then sterilize the loop again and streak on the surface of agar in the section C. After that, sterilize the loop one more time and streak on the surface of agar in the section D. Before settling down the loop, make sure to flame it again to prevent the bacteria from spreading to the environment and infecting the investigators. Next, incubate the agar plate at 37 degrees Celsius. The reason why the plate is incubated at 37 degrees Celsius is because it is the same as human's body temperature, thus allowing the quick growth and identification. Then replace the lid of the petri dish as soon as possible and secure with tape. This is because the lid will stop the additional unwanted bacteria in the air from contaminating the plate and make sure to not fully seal the lid, as this will stop oxygen reaching the bacteria, and this may encourage harmful anaerobic bacteria to grow. Lastly, after a day of incubation, pick up a single colony of each species of bacteria from the incubated streak plate aseptically and do streaking on the new agar nutrient plates to obtain a pure culture of each species of bacteria. So next I will talk about the precaution. Firstly, make sure to wash hands with soap and wear the gloves before handling the procedures. This is to prevent the investigators from catching any harmful bacteria and to avoid infection. Secondly is flaming the loop, which is by holding the loop in the flame of the Bunsen burner until it is blown into red hot for a few seconds. It will kill all the contaminated organisms and also sterilizing the loop. Thirdly, make sure to cool the loop before picking up the organisms from the inoculum culture. The loop can be cooled by letting it to touch on the very edge of agar before contacting the inoculum. The reason why the loop needs to be cooled first is to avoid killing the microorganisms that is in the culture. Fourthly, when using the loop, make sure to make a very lightly streak over the surface of an agar in a zigzag formation. This is to avoid from breaking the surface of an agar. Lastly, make sure to replace the lid to the agar plate as soon as possible to prevent the unwanted bacteria from contaminating the culture. So the conclusion is the aseptic technique is a basic laboratory technique that should be applied or employed, especially during the microbiology laboratory session, because it helps to prevent any contamination of the culture by unwanted or harmful microorganisms inherent from the environment that also can affect the accuracy of the result. So that's all from me. Thank you.